All right, so you should have already finished your first research project, which is one that I essentially just designed for you, that you would carry out, just to get an idea about setting up an experiment, collecting the data, some ideas about controls and questions that you would ask. And in that first experiment, you took little Ziploc bags, and inside the bags you placed peas, little seeds, okay, that would grow potentially a pea plant. And then we had a question, and actually in this particular one it became a little bit more complicated in that there were two questions. Right? So initially the question was going to be about soil. Does a seed need soil to germinate, to have the new plant embryo start to develop and then grow into an actual plant? Problem with that is that I realized was that depending on the soil that you had, it could have a very different water content. And so knowing that water is going to be potentially even more important than just soil itself, so the water in the soil, kind of split it up into two questions about water and soil. So you essentially had some little bags that had just a dry piece of paper towel and a seed. So no water, no soil. You had some with a piece of paper towel and you added water to it, some with soil but no water, and then some with soil and water. And then at the end of several days, for me it only took about 4 days to see some germination. And you were just scoring them, yes or no, did they germinate or not over a period of time. And then you would just end with a total number that germinated in just the paper towel alone, the total number that germinated in paper towel plus water, and then in the end collect the data and compare it to kind of see. Probably what you would see is nothing happens in the one with just the paper towel and no water. The ones that add water, pretty much all of them should be like 100%. The ones with the soils, we're going to see a lot of differences depending on the soil that you started with. So if the soil you started with already had a fairly high moisture content, you may have seen germination. If it was very dry, you may not have. But the ones with soil and water should have also been very high. So that's kind of the idea here. And you could start to see that there are problems or complexities in the design of an experiment, things you might not think about initially that then you have to think about. So that brings us to project 2. What is this? You now have to design your project. You will ask the question, you will pose a hypothesis, you will set it up. So I'm just going to give you some guidelines on how to do that, but it is not restricted to any one thing. You do not have to do pea plants again, although this will be much easier for you if you've already done that for experiment 1. You already have the seeds, you already have some experience germinating them, so that would be a logical place to go. In addition, there's cost. So how much will it cost for you to do? If you already have a whole bag full of seeds and you can still use them, you already purchased some potting soil, then you're just going to go ahead with that. You don't have to do anything else really except maybe some containers for it. Or if you choose to do something special with light or quality of light or duration of light or something special like that, or different temperatures that you have to change or alter the environment, then you might have to do something different, buy a special bulb or something for that. But that's up to you. That's your choice. You don't have to do any particular thing. So I'm going to give you this research project sheet. You'll have it on your site, and it's going to give you overall descriptions of the question, the hypothesis, the design. The design is going to have several parts to it. So kind of a part 1, talking about the treatments. Okay, so what are you going to do? And then part 2, the controls. So variables that you need to think about, that you need to kind of control for. Right? And then you have to come up with materials and methods, things you actually need. So you should write this down before you start, because you'll start doing it and be like, oh gosh, I don't have the containers, or I don't have something else, I need to go get that. So you make a list of those things. And then the data collection. So before you actually get it set up, you should think, well, what am I going to do? Like this one was germinate or not germinate. The plants emerged from the seed or they did not. And that was it, you can essentially say. The second one maybe now is growth. Okay, so you actually will germinate plants, maybe you take about 4 days to do that. Then you can plant them into soil and then start to come up with different questions, which I'll talk about in a second, ideas for that. And then you will maybe look at growth overall under different conditions. So now maybe you're going to measure the height of the plant. So you have to figure out, am I measuring the height? How am I going to measure the height? Centimeters, inches, what am I going to use? Do I have a ruler? Do we even have one to do this with? So these types of things you have to think about. Am I going to weigh it? Am I going to count the number of leaves that emerge? Maybe some have more leaves than other leaves. Some plants have more leaves than others. So maybe that's what you'll do. There's things to think about. What type of data will you collect? So you should think about all this before you get started. And then there's going to be a sheet for you to fill out. All right, which you probably can't read this in this video, but it's going to just be a blank sheet like this, not with all this typing on it. And you're going to fill in your research question, your hypothesis, the design, and there's a couple like points. Fill this in, fill this in, fill this in, and so on. Data collection, fill this in. So there are going to be two parts of a worksheet to turn in. So you're going to turn in to me part one. That's going to be almost immediately. Like soon you're going to come up with the idea within a few days, kind of get the question, how you're going to collect the data, what's your design, what are you going to do, and then you're just going to turn that in. Then you're actually going to do the experiment, and that could take 2 weeks, 3 weeks. It could take a while to get your actual data. Okay, and then when you're done, then you're going to turn in a second sheet which has some conclusions to it. Did you know your data, a data table, a graphical representation of the data, and then some questions about that you'll answer about conclusions. You know, did it support the hypothesis? Did it not? What were the problems with it? And then you'll propose research project 3, which will essentially be this didn't go well. I did this poorly. So here's what I'm going to fix. This is what I'm going to do better. Or here's what I'm going to add a question to this. I'm going to be doing two things at once. And then you're essentially going to redo the experiment. So project 3 will be your project 2 expanded or improved in some way. All right. So, and you're going to repeat it. So you're not going to have to do something totally different for that. This right now is going to be something a little bit different. It's not the germination of the seeds. It's something else. Okay. So the question, usually based on observation. Ideally, it's something that you observe in nature. Something happens, and you go, "Oh, I wonder why that happens." And then you could look into it. But the thing is, instead of just doing a whole research project, first you look it up. See, do people know this? You know, already. Typically in science, that's what people will do: is see, is there anything published on this already? And I'm just going to repeat exactly what someone else did. That's not necessarily bad, even in the regular scientific community. And there are actually new journals where people repeat exact experiments that other people did to see if they actually are repeatable, because science should be repeatable. You should be able to do the exact same experiment another person did and get the exact same results. And if you don't, either you did something wrong or they did something wrong. And it's a way, but if you get the same result, it's not a bad thing. It's a good thing that actually shows, yeah, we trust this information, and maybe then you take that and move forward and do something different with it. So that's something you could do. So in this class, it's just an intro class. So you're kind of just getting the feel for what is science and how do you ask questions and pose hypotheses. It's not meant to be a master's project or something like that. It's just an introduction to all this. So you're just going to ask a question. If it's been asked before, that is completely fine, right? And you're going to have it with your species, you know, so you have to know with your organism. So if it's the pea plant, then that's what you're going to do, but you might choose a different species of plant, or you could compare it between two different species. So it could be maybe this species would germinate into dry conditions. Well, you can do that step again with different types of seeds. So first you got to come up with a question, and this could be, you know, based on something. And let's just say it's growth. Okay, so plant growth, because you probably have seen this in a yard or a garden. One plant seems to grow really well, another one doesn't. Now, I keep talking about plants, and that's because that's what we've been doing here. As an organismal class, you could use another organism. Animals are going to be way more difficult to do, way more expensive to do. Say you want to do something with fish. Well, you're going to need to have an aquarium for each replicate. So if you want to have something with 10 fish, well, you need 10 aquaria, then 1 fish per aquaria. So think about that in terms of your design and setup. It starts to become really difficult. Having a little paper, you know, Dixie cup type thing with some soil in it for a plant is very easy and very inexpensive to do. And afterward, you could put it in the garden. After the research with the animal, what are you going to do with them? You can't just ignore them. So you have to figure out what to do. It's much more complicated to care for things in afterward. So you could put this in a garden, you could give it to someone else as a gift at the end. So the plants are much easier, I think, to manage overall and less expensive. So that's my suggestion. But again, the question is up to you. As long as it's reasonable and you can actually do the experiment, then that's fine. So don't feel like you're restricted to do this. But let's just say I'm using an example to try to explain this. Let's say you're looking at plant growth, okay, and we see, maybe these plants grow in the shade and these grow in the sun. So light effect. All right, so what is the effect of light on growth? And again, now you can start to get even more detailed in this. So just duration of light. 24 hours of light, 24 hours of dark. Extremes. That's one way to go. And that's often a way people start off, maybe with this project, where it might be something like that. Let the plants germinate so they're all the same. Then you plant them. Some of them get moved to a place, you turn on a lamp, it stays on all day, and after a week, you measure them, or after 2 weeks, you measure them and see. Another group kind of goes into the dark, and you kind of see what happens with those. They already germinated, they were the same, but will they grow in the dark? Will they not grow in the dark? Will they grow slower in the dark? Does it have no effect on them? That's kind of what you're going to try to answer. It could be intensity of light. So this is something a little more difficult. Maybe you could use this with different bulbs. So first you have to put them in environments that are controlled. So a dark room that's never going to get light. And then you separate it into two areas. One, you put a very bright bulb. One, you put a very dim bulb. So now they are getting light, and maybe you put them on a timer. So they're 12 hours on, 12 hours off. They get the same amount of light, but you have different intensities of light. So that could be it. You could have different colors of light, a red bulb and a blue bulb, and you have different, so wavelength of light is the factor. So it can be very complicated. But again, those things could be your next project, project 3, where you then parse it out into something a little more detailed. But in the beginning, my suggestion is start off with something simple and straightforward, like 24 hours of light, 24 hours of dark. I'm going to water these. I'm not going to water these, or I'm going to water these once in a week, and I'm going to water these every day. So you could have, watering, light, other sorts of things. Temperature, if you have areas that are warmer or cooler that are consistent. That's the thing. I'll talk about variables. It's got to be consistent. It can't just be changing randomly over time. But that's not going to work for an experiment. So then you pose your actual hypothesis, okay? So, which is what you think. That the plants in the light will grow more so than the ones in the dark, okay? So plants receiving light will grow more, and then by more, that's something we have to define, than plants in dark. Okay. Now, by more, again, that's going to come to the design and the data collection part of it. Like, what is that height? Again, you have to, or number of leaves, or you're going to weigh the mass afterward and see like how much is mass they built, because it could be thickness of the stem. There's all sorts of different things that could be defined as growth. Okay, so keep this in mind that you may have to think a little harder about exactly what it is you're going to measure as growing more, than another one. So if you pose your hypothesis, and maybe it's water, one's receiving a certain amount of water are going to grow more, one's receiving this, that's going to be the hypothesis. Then you're going to test it. So then it comes to your design. So things to think about. You should just have one variable that you alter. All right, this is your treatment. Just light, just water, just heat, just something specific, not multiple things. You can't have one in a warmer environment and a bright environment and water it more and then do this or combine them. You could do those things in a very big sort of complex experiment, but that's again not what we're doing. We're picking one thing, everything else has to be the same. So now this is important here. All other variables must be the same, meaning same soil, same type of pot or container that you put the soil in, same type of plant seed. if they're germinating, you have to let them all germinate and to be at the same starting point when you put them into the soil to begin your experiment. So they're already pre-germinated plants and they're all the same size, and then you start your experiment with them. There can't be some that are already starting off much bigger than others that you put in bias one treatment versus another. Everything has to be the same when you start it off. Then when the experiment continues, same thing. Natural daylight is going to affect the experiment. So if you have it by a window, that's going to throw things off if you are testing light, because the actual daylight on a bright day or a dark day is going to change how much light they're actually getting. Even if you're turning on a lamp next to them, that's probably going to be insignificant compared to the natural daylight. So you need to control for the environment when you're doing this, and you need to do this in a space that you can actually control that environment. So keep that in mind, that everything has to be the same. If you're watering them, you have to measure out exactly in milliliters how much you're giving them, and it needs to be happening at a certain period of time that you put into your design. So you're watering them every day with 5 milliliters or 10 milliliters or what you're doing. And then that's going to be the part of your design. Okay, so you have to have control. So you have to control these variables, and then you have the test one that your treatment that you're actually doing, so that you're altering the water, maybe amount of water, you're altering the amount of light, but everything else has to be the same. And then you come to data collection. So when it comes to data collection, like I said, you have to decide ahead of time, what will you measure? And record. How will you measure it? Do you need a scale to do it? So I'm going to weigh them. Well, do you have a balance? Do you have a way of measuring them? Or if not, then maybe you can't do that. So you have to think about what you can actually do that's realistic. So can you count the number of leaves? Yeah. Can you probably get a ruler to kind of measure the height? Yes. That's probably very easy to do and very inexpensive if you don't have one to get one. Well, there's other things that might be difficult to obtain, and if you don't have that or have access to it, it might not really be possible to do. So then we come back to your question. That's not a question you can answer or a question you can test right now, because you won't have the equipment. That's why when you go into the design, after you kind of do this, you do have to kind of list your methods, or list your materials. Because at that point you may find out, oh, I don't have this, or I need to get it, or I just can't get it, and I need to go back and redo my question. So what you're going to turn in, hopefully within the next few days, all right, of starting this project, is just a question. Just decide on it. One sentence, pose your hypothesis. That's it. Then you have to think about the design. How are you going to set it up? In addition, you have to think about your replicates, right? 10 fish in one tank is technically an N of 1, so they're all in the same tank. They can interact with each other. They might fight with one another. They might compete for food. It's not really the same thing as having 10 fish in 10 separate tanks. Then each one is an individual that is part of your study, and so that would just be the one group. And then if you had a second group, a control and a treatment, or 2 different treatments, 2 different types of food, or 2 different amounts of food, then you need 10 other fish in the other group, and that sort of thing. So in this case, we're doing with the plant, so maybe you have 10 little pots. Times 10. And that gets, 24 hours of light. And then you have 10 more pots, and these have little plants in them. And they're in 24 hours of darkness, okay? That's, like I said, that's an extreme. You probably may not want to do that. You might want to give them some light, 2 hours or 3 hours, something like that. But again, it's up to you. And whatever happens in this experiment, you'll modify it in the next one. So if things go poorly with this one, that is fine. You will have learned something, and then you'll use that to refine your design for the next project, okay? But you should need to have more than one. You can't just have one member of each group. That just won't work, and you have to redo the assignment. It won't count, essentially. So there's certain things that if they don't work out, that's absolutely fine. That's part of doing science. Most of the time, things don't work out and you have to redo them. You have to redo them many, many times to even just get an experiment to collect data from. Not necessarily that it works the way you want it to. It's not supposed to work the way you want it to. You're not supposed to want it to work anyway. You're supposed to want to just help answer a question, and you're designing something in a non-biased way to try and answer that question. Right? That's what it's supposed to be. All right. So, but if you just don't set up things with certain criteria, like having enough replicates, enough numbers of individuals in a group, then the results you get are just kind of meaningless. So you need to have quite a few. Depending on what it is you're doing, it might be very difficult to manage or expensive, and then you might have a smaller group, with 3 to 5 individuals in it. Whereas if it's very inexpensive and easy to work with, you might expand it. There does come a point where do you need 100 in each group? Probably not. You may not get any more or better information than if you had 20 individuals or something like that. 100 versus 20 maybe doesn't give you any difference in sort of the power of your data. So there are ways of calculating that in statistics, the power and finding out how many you need. Again, that's a biostatistics course, something different. We're not really getting into it. So collect your data, figure out before, you're not going to collect it now. You're just going to propose how you're going to collect it, and then that's it. So that's the part 1 is just all this sort of proposal part. All right, so the proposal includes your question, your hypothesis, your design, and how you're going to collect your data. That's it, and you turn that in. Part 2 is going to be the results. And then after that, then you're going to very quickly, you'll just turn over project 3 pretty shortly after you finish project 2, because, like I said, it's simply going to be an extension of this project, or not a duplication of it, but you're going to repeat it with something altered, or you're going to improve something, you're going to change the variables in some way, you're going to change the number of replicates you do, you're going to fine tune the light, you're going to go to a different wavelength or a different intensity, you're just going to do something that's in the same question overall. Like I said, you're just going to kind of focus in on one area of it, or just improve the study. So hopefully that along with what's written on the general written explanation of the experiment is what can get you started on turning in your part 1, the proposal, and then actually getting your whole project set up and to get working on it.